This is a presentation about the model to simulate underwater visual surveys of fish populations. And what are underwater visual surveys, or underwater visual sensors as they are called? These are basically methods to count fish underwater and estimate the size of a population. They are non-destructive and cost-effective methods used worldwide in conservation management experiments. They basically involve a diver moving along an area or staying on a fixed point and counting fish and then estimating the density of fish, and this can be achieved using a huge variety of methods for different radius of observation, different widths and lengths. For instance, in a strip transect, the diver can move along in a fixed direction and look at a fixed width, and this can be wider or narrower, for instance, longer, shorter. Or if the diver stays in a fixed point, it can have a larger radius, stay there for more or less time. And this is a very difficult thing to choose, because usually you don't there's no objective way of choosing one width over another or one length over another, and this is usually a choice made based on tradition or a common practice on a certain institution or research team. When using any sampling method, we are faced with mainly two types of errors: random errors and systematic errors. And random errors are mostly linked with precision, or the dispersion of our estimates to both sides of the mean, and accuracy, or the deviation from the true, in this case from the true abundance of fish. And while precision can be quantified by repetition and minimized by increasing effort, accuracy is very hard to detect and minimize. We can have a very precise method but be completely wrong and completely unaware that we are wrong. And this has been shown to have a strong link to species behavior in case of many of these visual senses, because fish react differently. Different fish can react differently to an observer. Some fish are shy, some fish are curious, and this will generate systematic errors. And this is where models are useful. Some other models in the past have approached this problem. The Reflex model in 1995 used the grid-based movement with a 10-second time step and looked at bias due to fish speed and approach angle. So how fish coming on different angles towards the diver would generate different biases. And the Anindens model in 2010 used the correlated random walk and looked, using a much smaller time step, looked at how the fish speed relative to the observer can sometimes generate overestimation, because we are doing a non-instantaneous sample. We are not taking a snapshot. We are scanning an area and gradually moving along an area while new fish are coming in the sample, and this generates great overestimations. But none of these models featured very complex behaviors, which has been pointed out as one of the factors affecting accuracy in these methods. And this is where this model comes in. The fish senses model is especially explicit to the individual based model written in ETLOGO, and it applies a vector-based movement like a Boyd's movement algorithm with a much smaller time step. Basically each fish can have up to 8 normalized vectors, and all the vectors are summed to generate an acceleration vector that calculates the velocity on the next time step. And these vectors can be multiplied by weight coefficients, and these weight coefficients are fixed and define behaviors. So by shaping the length of the different vectors, we can shape different behaviors. While, for instance, a searching behavior could have a wandering vector, a vector that makes fish wander randomly, would be very important in a searching behavior. A more directed vector towards the patch would be more important in a feeding behavior, for instance. Up to 4 different types of behaviors can be defined per species in the model, and we can define behavior frequencies, and every 10 seconds this interval can be is arbitrary because there's no external influence on behavior, but this is roughly based on what was used by Watson and colleagues on the reflex model. Every 10 seconds we get a random pick from the pool of behaviors with the given probabilities. So in the model, for the sampling model, we have several parameters. We can choose the method, stationary or strip transects, with all their different measures. We can have fixed view angles for the divers, a limit for underwater visibility. Counts occur every second. This is mostly for performance reasons. There's no need of counting every tenth of a second, because fish that are counted are not immediately recounted. They go to a memory list, and I will talk about that in a minute. There is a count saturation of 3 fish per second, because it's not humanly possible to count 100, 200, 300 fish. Even if they're in front of us, it's very difficult to count all of them in 1 second. Priority is given to the closest fish, and there's a memory, as I told before, and when fish leave this cone of vision defined by view angle and water visibility, if they leave this this cone, they are forgotten, and if they come back in, they may be recounted. There's actually a maximum ID distance that's specific to each fish species or each type. Some species are easily recognizable even when very far away, some others are not, so this is actually an important factor. As for the fish parameters, there are some parameters related to drag forces, such as their size, their length-weight relationship, their length-surface area relationship, and their cruise speed and burst speeds. This should be taken from the literature where available, or lab experiments. There's perception angle, perception distance, if they are very approachable, or if they are scared from even from long distances. Detectability, if they tend to be cryptic and hide in caves or mimic their environment. This is a probability of being missed. And of course the list of behaviors, up to 4, and their frequencies. And the parameters and the weight of the vectors should be shaped, and they can be shaped in real time, as we'll see in a minute. And the effect of this shaping should match observed patterns from, and for instance, underwater video, as we are seeing here. And this is the transition from a searching behavior to a feeding behavior in this schooling species. Some applications of the model are, for instance, the calculation of correction factors for field data, because we don't know the true abundance in field data, but we can calculate bias in the model and then correct the field data and get a more hopefully a better picture of what should really be there. Some other applications are looking at the effect of sampling design, how varying widths, speeds, distances, radius can affect inaccuracy or precision. We can use this for power analysis. We can use this for method optimization using search algorithms, trying to get a better combination of method parameters for a given species, or even to look at community assessments. This is quite performance hungry, but we can actually upload more than one species and look at the effect of having to count multiple species at once, or applying single species assessments. The model is available in OpenABM, and a lot can be found there, including the citation to the original publication. It's model 5305. There's a project in ResearchGate as well, and a little section on my website. And I will now look at or show you the model interface. Here is the interface for the species creator, and here is the actual model. There are actually 2 parts to this model. The species creator is where we basically, as the name implies, create the species. I will click here, create new species, press setup and go. I will speed this up. In this case we have a schooling species. As we can see, these are the weights of the different vectors: wandering, resting, cruising, prey chasing, spacing center, space center, and a line you may recognize from the Boyd's algorithm by Craig Reynolds. And this defines basically the schooling behavior. And if I just switch schooling off here, all three vectors get disabled, and we get the dispersion of the schools. So we can have schooling species or solitary species. We can actually deploy divers to see how fish react to divers, and if the patterns are matching what we know from the field or from video data. Here we can, for instance, change the approach distance. In this case 1.5 meters. I can increase this, so I will have fish reacting to the diver earlier from a longer distance. In this case the diver avoidance weight is positive, so we get avoidance. If we can set this vector to negative, we get attraction. So basically now fish should I should increase a bit. Fish should feel attracted to the diver, and as you can see here, these schools are slightly being attracted to the diver here. And then we can of course change here, save up to 4 behaviors in these slots, and then save the species data to a file. And this is important because if I work a lot with a given species and I know it well, I may just model it here, export it to a file, and it can be used by someone else directly without having to copy individual attributes or create the whole species from scratch. And then let's imagine I would export this species with a given file name. I'll just stop here. If I would export this species with a given file name, for instance example.csv, I can just input here the CSV file name, and this is the actual model where the count occurs. And I can choose the method here from fixed time, fixed distance transect, stationary point counts, and random paths. In this case I can put a fixed distance transect, for instance, and I will go here to fixed distance transect parameters, choose a swim speed 10 meters per minute, choose a width 2 meters, and choose a distance, for instance 15 meters or 10 meters. Let's make it 10 meters. So this is short. I can define max visibility, count saturation. I can define the initial time to stabilize movement. When I press setup for the first time, the movement will stabilize for a certain number of, because the placement of fish is random, so I don't want to start with this abnormal dispersion of fish. So when I press setup, in this case the fish movement will stabilize for 20 model seconds. Schools will start to form because this is a schooling species. We get these little groups, and the diver is placed down here. And I can now press go, and as we can see, the diver is now performing the counts. And I will now shut down this view, shut off the view updates, so I can speed this up. The model is a bit performance hungry. As you can probably see here, the seconds are almost real time, just a tiny bit faster. And as the diver is counting, we can probably look at their if you can see here, we can look at the memory. There's the memory list, the counted fishes. In this case, they're all named schooling. If we had different species, their different names would appear here, and then we could count how many of each type. And I think the diver stopped because it's over, and we get here the density estimates. We had a true density of 0.3 fish per square meter, which makes a total of 480 fish in this whole area. And here we get actually the bias due to non-instantaneous sampling, which is compared with a snapshot. So when the diver starts, this takes a snapshot of the transect he's about to do, and then it does the count, and then the bias is calculated here. And in this case, he estimated 0.5 fish per square meter, so it was a 0.2 difference from the. It was an overestimation by 0.2 fish per square meter in this case. And so this is an overview of what the model can do. And for any questions you have, go ahead. And thank you.